Welcome to the LowCarbMD podcast. No one is beyond help. No one is beyond hope.
As we've always said, we're bringing you medical information and cutting-edge science, but none of this is medical advice. Please seek out input from your own doctor. Guys, welcome to the LowCarbMD podcast. I have an amazing guest, somebody who I've been very interested in. I've followed on social media. I've watched lecture. I've read his publications in the New England Journal and JAMA, Lancet, and elsewhere. Dr. Boulware, he's an infectious disease doctor. He went to medical school in Indiana University, residency and fellowship at the University of Minnesota, and he is a professor of medicine as well as a professor of epidemiology. And you are at University of Minnesota, right? That's where that's where you teach and do your research.
Indeed, Minnesota. You know, it's a place where if you don't like cold weather, then you probably have never been here. But you come in August, you fall in love. It's a great place. By the time November rolls around, you realize you made a terrible mistake.
So I want to, first of all, I'm humbled that you've come on board. We've had some infectious disease doctors come on board and kind of set the record straight. And one of the things that I really want to focus in on and maybe start in on was, you know, the PTSD we've all had over the past 2 years. And I know as an infectious disease doctor, the amount of pressure that's probably been put on you has been immense. One of the things that really impressed me, you know, I said this off the air, I've been following the FDA regulatory environment for over 20 years, even before med school. You know, I analyzed advisory committees for hedge funds, and I have never once in my career seen a band of doctors get together and file an an application with the FDA for a drug. And that drug was Fluvoximin, you know, for, I guess the indication was preventing severe, COVID severity. I've never seen this. So the minute I saw this, I was like, who is this guy? What is he about? I want to know everything about him and how he came to that moment. So you know what, I guess maybe we could start about what brought you to medicine, just to go way, way back, how you got into infectious disease and public health, and then how did this all unfold? And most recently, you wrote 2 articles. You wrote an article in the New England, no, it was in Lancet, I believe, you know, basically saying the nail in the coffin for hydroxychloroquine. You just published the Act 6, and that followed ITech, which I think are the nails in the coffin for Ivermectin. So how did we get here?
How did I get here? Well, I don't know if my story's that interesting. You know, I, back in college, you know, I was sort of deciding between sort of basic science, sort of career paths of chemistry or physics or something like that, and medicine. And I was sort of I was out for a run and was running through a sort of a cow pasture one day in sort of rural England. And I realized, like, I really like people. And so versus like a bench scientist, and so I was like, I think I want to go to med school. And so it's like one of these profound moments that sort of shapes your career. And during med school, my first 2 years, I was in beautiful Gary, Indiana, which is one of the branch campuses for Indiana University. And, by summer, I wanted to get the heck out of Gary, Indiana. And so I went hiking on the Appalachian Trail, and I hiked about 1,000 miles on the Appalachian Trail. And as part of that, I did this survey of Appalachian Trail backpackers. And so it was a prospective cohort study of what injuries and illnesses they had. And from that, basically, that was kind of how I got into research, of kind of a research, I was like asking a question and trying to answer it. And followed on a bunch of kind of fun sort of wilderness medicine and travel medicine kind of studies and small clinical trials. And eventually I decided, well, I kind of like this, the infectious disease and sort of the tropical medicine components of medicine. And so that's how I ended up to be an ID doc.
So you're like the hippie of infectious disease doctors, right?
I'm not sure I'd quite go that far, but I certainly did a lot of wilderness medicine stuff before I was working on HIV. But I realized wilderness medicine, can't really do an academic career based on that. It's really hard to do that. Versus HIV is sort of a little bit more of a standard career path.
So you get into research. Eventually, you get an MPH. Was that right out of the gate, or did that come on later?
It was during my infectious disease fellowship training. I got an MPH along the way. I did a bunch of, sort of self-taught statistics, through med school, and not really well done. And one time I sent this, it was a randomized controlled trial on jellyfish sting inhibitors. And I sent it to JAMA because I thought, wow, this is a great study. And so one of the critiques is like, this person clearly doesn't know anything about statistics, but it's a good trial, but they need to learn something about statistics and get a statistician, basically. And so that kind of prompted me to say, maybe I should actually get some formal training in epi and stats. And so I did so during my fellowship.
Got it. So you get training, and you have a passion for infectious disease, and it sounds like, passion for clinical trials and research. And, before COVID, so pre-COVID, if you can, remember those times, where was your passion? What did your career look like? And, what are you doing?
Yeah. So in the ID world, like pre-COVID, like my focus was actually on meningitis. And so I've worked in Sub-Saharan Africa, particularly in Uganda, for not quite 2 decades, but pretty close, since 2005, and particularly with cryptococcal meningitis and TB meningitis. And so we've done a number of clinical trials, and being sort of a neglected disease, like we've got to do everything. There's no drug company that's doing all the paperwork. Like we're filing with the FDA, we're doing sort of all the, all of the work on those trials. And so I learned how to do clinical trials with some mentorship, but basically, yeah, my expertise is known as cryptococcal meningitis, which is pretty far away from COVID.
Yeah. So, but it sounds like you're doing a lot of the grunt work outside of industry, right? So that maybe that's a caveat, because people with an MPH, MD background with an interest in clinical trials, majority work for either a big institution, government, or pharma, an industry, I should say. So it sounds like
that's very true. And I think it's particularly in infectious diseases, most of the clinical trials are either run through networks or they're pharma sponsored trials. And so we're one of the few that's really had an investigator initiated NIH grants for clinical trials, because otherwise most of them are run through networks. And so that's kind of unique and I guess has paid dividends for NIH down the road.
So, okay, here you are, kind of really sounds like outside of the quote unquote industry, outside of the system, kind of pursuing your passions and following your calling, getting yourself more and more education, and then COVID hits. So how does what happened, February of 2020 for you in terms of your career?
Yeah, so January, I think like with everyone, it's like, oh, it's kind of interesting. It's in China, like, not really paying attention to it. February, like, still not super interested. And then when they're starting to have community spread in the U.S., you realize like, this is gonna be bad, and it's gonna take off. And so we were supposed to my whole team was supposed to go to an HIV conference in early March, and I just gotten back from an NIH study section, and in the HIV meeting, the CROI meeting was canceled. And so we were supposed to go to Boston, and we had like 4 or 5 days on our schedule that were like blank all of a sudden. And so I was like, we should maybe do something about this. And so I emailed some contacts at NIH and who had worked with on Ebola trials and things like that in the past, and kind of asked like, what's NIH doing about outpatient, about prevention or about early treatment? And so at the time, NIH was really focused on getting their Act 1 study, the Remdesivir trial, up and running, and there was hospitalized patients. So at the time, they had no plans for outpatient treatment or post-exposure prophylaxis. And so we thought, hey, this is going to be the key of like trying to kind of block infection, stop the transmission of infection. And as a healthcare worker, for my friends who are being exposed, the recommendation was, just go home and quarantine for 10 to 14 days. I was like, well, can we do something better than that? That's kind of like, 14th century technology from the bubonic plague. And can we do something better than that? And so that's how we kind of said, well, let's do a post-exposure prophylaxis trial. And so I'd worked with hydroxychloroquine before on some HIV studies. And so I knew a little bit about it, and from a tropical medicine angle, I was familiar with chloroquine and hydroxychloroquine. And so we said, well, let's, we knew there was some in vitro activity from SARS-1. And so we said, let's do a post-exposure prophylaxis trial. And so I emailed my team and said, hey, let's get together at 8 o'clock on Monday morning, emailed them on Sunday night. We're going to do a trial. And then said, let's come together and let's get it done. And so sort of March 9th, we got together and said, look, we're going to do a post-exposure prophylaxis trial. And my team was like, okay, sure, let's do it. And we kind of jumped on board. We wrote a protocol in about 48 hours, submitted to NIH, submitted to our IRB within about 48 hours, submitted to FDA within about 72 hours, built a database, sort of, FDA kind
like
of
red cap or whatever, something like that.
Yeah, it was red cap database kind of thing that we'd done before. And we kind of got this up and running in about 8 days in total.
That's insane. That is absolutely insane. Like, people don't understand, like, this is something that can take months sometimes.
For years. I mean, I got this idea and let's get funding, let's do this and write a protocol. And we got IRBs and FDA and all the regulatory hurdles, institutional hurdles. So 8 days was crazy fast at the time.
So you're running this trial for hydroxychloroquine very early. There's some mechanistic data that says, okay, hey, this may work. Now, we know what happens next. Some faulty data gets published in the Lancet, New England Journal, JAMA, ends up being retracted. I mean, from my angle, I'm looking at this like, okay, there's people studying this. There's politicians saying that we should be inhaling hydroxychloroquine. There's, how do I make sense? Like, how do these studies get published and then retracted? And what should I think, right? What should I think?
Yeah, at the time there was the French data that had come out right around that time as well, and then there was rapid, politicians that were endorsing this. And my whole thing is, I don't know if it's going to work, but I know it should be tested, and that we should do a randomized, double-blind, placebo-controlled trial to figure out, does this have any benefit or not? And so all of these cases, we're going to have an observational cohort study or this or that, like all of that is noise. And ultimately, you need to do a randomized trial to figure out, does it work or not? Because much of the data early on was really junk data, and it was hard to really make any strong conclusions about.
But so, how does it occur, right? So you mentioned that NIH was working on Remdesivir. I mean, this is how my brain works, and maybe I'm just conspiratorial, but, we knew Remdesivir was kind of like the preferred, the preferred approach early on, right? It got tested, it's up and running very early on. And then we see, this phantom company just shoot up, publish a whole bunch of stuff, and then, it stifles a lot of research that may or may not have happened, and then it gets retracted. I mean, what do you think when you see that? Is that nothing for you? It's like, look, this is just taking me away from my focus, which is a trial needs to be done. I mean, what do you think when you see that?
Yeah, I remember it was like right before Memorial Day weekend or something when all that came out. And I got called by someone from CNN and said, oh, can you look at this that just came out? And I was like, okay, I'm headed out the door in an hour. But let me read this actually and try to figure out what the hell's going on. Maybe Jack edit that out. So I was trying to, so I was asked to like figure out like, what is this? And so I looked at it quickly, and some of it just didn't make a whole lot of sense of like they're extracting all these electronic medical records in Africa, and it's like, where are these places in Africa? And so some of it just didn't make a whole lot of sense. And then I noticed like that was actually the Surrogate thing was actually when Ivermectin first sort of came up as a thing, because they had a benefit for Ivermectin. And so it was a little weird, I guess. Was kind of my initial, like, I was a little skeptical of it. And once again, it's sort of a case control study, which has limitations already. But yeah, the fact it was like this worldwide sort of cohort and electronic databases and, from places in Africa including places in Africa, didn't make a whole lot of sense. And so there was some skepticism, I think, that rapidly was brought on.
But, our colleagues, right, at prominent journals weren't as skeptical, I guess, right?
Well, I think one of the things with the, medicine is built on trust. So like, who would like try to publish a high profile, totally fraudulent paper? Like, that's ridiculous. Like who would try to do that? And so, because you generally be caught. And so but it's strange times. And so people want to be famous or whatever. And or there's a profit margin of, oh, use my database. I don't know why they were, why they did that, but it is a weird thing because normally, people can make errors or you have typos. I mean, I've made typos on manuscripts or, people can make errors, but like to fraudulently put something forward that has like no data is kind of beyond the pale. And I guess the fact that there was some inherent trust that was built into the system, now there's a lot more skepticism, I think.
But you're plugging away. So you're seeing this kind of unfold and you're plugging away at that time on your clinical trial. You're probably, I don't know if you have prelim data at that point.
Well, the trial is over by that point. So,
got it. Okay. Yeah.
Yeah. So we started March 9th. Our first patient was enrolled March 17th and we were done May 3rd. And so by that point, by May 6th, we'd already written up 2 of our trials and sent them off to the New England Journal. So at that point, we already had our answer, and we kind of knew that there wasn't a big benefit. It didn't really work. So yeah, that was about I think the Surgifir stuff came out about a week before our first trial was published. So we kind of already knew our results. So
and the other thing that struck me as weird early on was I think it was the recovery group. I forget. I have to like go way back. There's so much PTSD to unravel. They really improperly dosed the hydroxychloroquine. I mean, in the trial, I forget which group this was. It was like,
it was the recovery trial. And so there was actually, I was part of these discussions because the question, we actually published a paper on this, like, what's the right dose to use? And so for my group, we just picked like, okay, what's the maximum FDA approved dose? Because we knew that if we went above that, then there'd be more side effects and FDA would never approve it, or there'd be a long discussion back and forth of like, well, it's safe, it's only gonna be for a couple of days. And so we knew like, okay, we want to get this approved quickly. And so we used the maximum dose that's basically used for malaria treatment. And so that's how we picked our dose. In the modeling studies, and we were part of some of the discussions about this, the modeling suggested, well, the doses probably aren't high enough. And so you did want to go a little bit further, to try to get this magic antiviral effect. It turns out all the dosing sort of things were based on the same paper, and that paper was wrong, that the doses actually, the concentrations in humans actually were lower than what they suggested. And it was a difference of whole blood versus plasma levels, and gets into the sort of the PK sort of details of weeds. But it turns out that paper was wrong. And so everyone else's like assumptions on what drug levels we'd actually reach in humans was also wrong. And it was way too low to be sort of an antiviral effect. Versus what you needed to actually achieve in humans in the in vitro test tube sort of situation versus what the levels you could achieve. And so so recovery did use a higher dose, but it was like 25% higher than my dose. The dose we used was safe, and nobody died from it. And so then like, oh, they used 1 more tablet a day. Like, oh my gosh, like they killed everyone because of the dosing. Like, no, that's not really the case. And so the people that picked the dose actually were really experienced in malaria and actually had used the drug for decades. And so actually knew quite a bit about the drug. And so I actually kind of trust their judgment. We didn't use that dose, but I don't think the dose was the problem.
It wasn't like so far off. Yeah. So basically, now
in the recovery, the dose in the recovery trial, there was a chloroquine trial in Brazil, which did use like 50% of the lethal dose. And if you run that for 2 weeks, my gosh, yeah, you do start to kill people. So that's a different dosing thing. But the hydroxychloroquine in the recovery, it was a high dose, but there were probably side effects, but it wasn't toxic.
So I mean, I'm like a lowly internist, and I'm trying to make sense of this, trying to take care of patients, and I'm seeing, okay, wait a second, Surgifir is retracted, maybe the dose isn't right. Politicians are saying this and that. I'm trying to make sense of all this, and here you are plugging away, actually doing the trials, and then it comes back really like it's a wash. Right?
Yeah. Didn't do a whole lot.
And at that point, are you saying to yourself, this is not worth testing anymore? Like, do you remember, April of 2020 saying, look, this isn't going to work. Likely we shouldn't waste any resources. Like, when was the first time you thought, we should ditch hydroxychloroquine?
It was after we got our results back. I mean, it was May 3rd when the trial was stopped and the DSMB revealed the results, and it's like, and it's like, this does not look very promising. We thought, well, what if we looked at this way and kind of, is there something we're missing? Because you don't want to be wrong. And so, we kind of were like, no, this doesn't, this is not very promising. And so, maybe, and the question then becomes, is there a zero effect or is there a minimal effect? And so you can kind of dance back and forth about, well, there was a 10% benefit or, and so we kind of realized by basically May 3rd that this was not really very beneficial. And we kind of, we sat on our, there was follow-up still ongoing of 2 weeks of follow-up from when we stopped enrollment. So sort of mid-May, and sort of the final analysis was late May before publication in early June. But we kind of knew it wasn't beneficial. And so I actually had gotten, this is a public record, but I had gotten emailed by the president's, personal physician, after he got exposed to COVID from his valet. And so he asked about hydroxychloroquine post-exposure prophylaxis, and someone referred him to me as sort of the expert on the topic. And so I didn't share our paper with them because they're not exactly I didn't share the paper, but I did tell him, like, I do not recommend him to use hydroxychloroquine because there's no data that it works. I didn't show him the data, but said, here's the dose we use, here's the side effect profile, so you're aware of it, it's safe.
I forget if he took it or not. I know he got antibodies.
Yeah.
He did take it. Okay.
So back in May, he took it. So I was, so we knew it. And then, people asked me, I told them what the results were confidentially, but not for public consumption at that time. Because I realized, like, rapidly, it had already become very political at that point. And it didn't matter what we said, like, people were going to do whatever they wanted to at that point.
Yeah, it feels like that's still going on right now. Okay, so then, all right, so you're doing the work. This is like the work that needs to be done. Turns out to be negative. And then, right around the time, I remember that's kind of when we started seeing the SSRI data. We started seeing the people getting, besides obesity, diabetes as huge risk factors, anxiety and stress-related kind of disorders are a huge risk factor for COVID severity. And then comes in some of the Fluvox data. I mean, is that when you started getting interested in Fluvox, or was it before?
A little bit. Like after my trial started, one of the people that was running the Fluvoximin trial out of WashU, she tweeted or posted on Facebook about, hey, they're running this Fluvoximin trial, refer patients. And so I, retweeted it. And at the time I thought, this is a dumb idea. Like, why is an SSRI going to work on COVID? Like, it's an SSRI. Like, why? Like, there's no antiviral effect. I don't understand. So when I first heard about it, I was like, this is a dumb trial and it's never going to work. Having said that, I actually did an SSRI trial for cryptococcal meningitis because sertraline, Zoloft, has in vitro antifungal activity. So I've done like a 500 person sertraline trial, which totally didn't work. Like, it worked in vitro, it had some other prelim data, but didn't work. And so I've done SSRI trials before, and it's like, this is a terrible idea. But they published it, and this is back in November of 2020. They published this small trial. It was 80 persons versus 72 persons, and it looked good. Like, they had basically, of the 80 people randomized to Fluvoximin, none of them clinically deteriorated to progress to severe COVID, were having hypoxia and shortness of breath and they're being hospitalized, versus there was 8 of 72 of the placebo group. And so it's like, like, this is statistically different, but it's a small trial, small study. It's hard to know what to make of that.
So kind of, and what's going on at that time with Ivermectin? I don't remember when it came into favor, right around the same time.
I think they were right around the same time of November, December. There was a hearings that the senator from Wisconsin sort of held, and so they talked about hydroxychloroquine. They talked about Ivermectin. So that was one of the first times that Ivermectin sort of made a public appearance.
Got it. Okay, so
right around that same time.
So Fluvox is coming in, Ivermectin's coming in.
Yeah.
I forget when the CDC or the FDA's famous social media post. I think it was right around December or somewhere around that time.
I thought it was later. I thought it was in the summer. I thought it was in mid.
Oh, was it? I don't remember. It's all PTSD for me.
Yeah, I think it was actually maybe June or July of 2021 with the horse thing.
Okay.
Because at the time, Active 6 was enrolling. So maybe, yeah. So maybe
So
jump
Active
back.
6, just so people don't, just people realize, Active 6 was a study looking specific. I think it looks at multiple repurposed medications, but a big arm was ivermectin, of which results were recently published, which basically showed that, you know, there may be 1 day less of symptoms with ivermectin, but not much else. Did I get it right? Or
basically, yeah. So Active 6 came about in sort of January of 2021. And actually sort of Janet Woodcock and others at FDA kind of admitted, like, one of the things the FDA has not done very well was early therapeutics. And so she kind of pushed, I think, behind the scenes and others within the new administration of like, hey, we should look at some of these repurposed drugs, because there was a lot of clamor about ivermectin, people were using it. And so let's actually study some of these repurposed drugs. And so with that, via sort of congressional funding, came out this Active 6 trial. It's a platform trial looking at multiple different medications. And so ivermectin is one of those. They're looking at steroid inhalers, looking at fluvoxamine, and there's other sort of things in the pipeline as well. And so it's a platform trial looking at some of these repurposed existing low-cost medications for which there's a ton of safety data, and people are using them. And so the question of, do they actually work?
Yeah, and that's important to mention, because, you know, during that time, post this initial hydroxychloroquine data, not only did we start to see Fluvox, you know, kind of penetrate into some clinical practice, we also started to see mouthwashes. We also started to see penetrate into some clinical protocols. Who knows if they work? But the recovery trial showed steroids work, I think, in the fall, right? That was
actually July of
2020.
2020.
Yeah. So now we have another. So now we have Remdesivir, emergency use approval. You know, recovery trial showing that steroids in severe disease help. And now we're repurposing budesonide and fluvoxamine and some other therapeutics and seeing if they work. Where are you in terms of your clinical practice? You know, at the, I guess, right around, that's when the Moderna, the vaccines were approved.
Yeah,
right?
December of 2020-ish. So yeah, after kind of our hydroxychloroquine trials, we did basically 3 trials, 2 on prevention, 1 on early treatment. Like, I got a bunch of social media just nastiness and hate mail and all sorts of just stuff. And it's like, just like turn off Twitter and like get back to my normal research work and like let other people work on this, because there was the monoclonal antibodies were under development and there was a lot of other stuff that was kind of in the works. And so I was like, that's fine. And I was happy to kind of go back to my normal meningitis work. And where we're still running trials in Africa while all this other COVID stuff is going on. And so I kind of got drawn back with fluvoxamine, because after that November thing came out, there was this outbreak at this horse racing track. So this ended up to be like a 60 Minutes piece. I don't know if you saw that, but it was basically Dr. David Boulware out in the Bay Area in California had read the paper and knew fluvoxamine, and there was basically this big outbreak among all these like workers at this horse racing track, and over 100 people. And so he offered some of them basically fluvoxamine, and some of them accepted, some of them declined. And so he had really good results, once again, of like everyone taking fluvoxamine did great, and then people who didn't, some of them got hospitalized, and one of them died, unfortunately. And so he had this data, and I was like, well, you should write it up. And he's like, well, you know, I'm kind of busy. And so I was like, well, I'll help you write it up. Like, if you've got the data, like, hey, let's write this up. And so we wrote it up as just a observational real world cohort, and published it in OFID. I think it was in early February of 2021. So I kind of got drawn back into the spectrum of COVID, and was sort of asked to help on a couple other clinical trials. One with one of my colleagues in Minnesota, who's testing metformin, which is one of your obesity drugs, which you might be very interested in. And then also was asked to be sort of one of the national co-chairs for this NIH Active 6 trial. And so while I was sort of trying to get out of COVID, I sort of got drawn back in. Via sort of
once you try to get out, you know, they just bring you back.
You never get out, yeah.
So where are you with antibodies at that point? You know, I was very favorable on the antibodies early on. You know, we had a huge population who was just, for whatever reason, good, bad, or personal values, whatever it is, very vaccine hesitant, and we were forced to really adopt antibodies early on. So how did you feel about those early on?
Yeah, I mean, I thought the monoclonals were great. One of my old-time friends now works for Regeneron, and so they had some really good data initially early on in the initial variants, and yeah, so the initial monoclonals were great and certainly life-saving therapies for high-risk people who were getting sick. And so that was that was I think one of the really first effective therapies. But it's as we all know, with each variant, we're kind of each time a variant pops up, we're losing different monoclonals, and so they're not as robust as some possible therapeutics.
So I think at that time, I remember feeling kind of disheartened by the regulatory process. I remember starting, right around the spring of 2021. There's the emergency use approval for the vaccines. I would say, I would have liked to see a little bit more robust data in terms of the length of the study and clinical endpoints. But, now we have something approved, right? Now we have something approved. How do you feel like, having been in clinical trials for a long time, right, about the speed at which our clinical practice changed, right, with regards to whether it was vaccines, whether it was ivermectin, hydroxychloroquine, antibodies, off-label uses? How did you feel about the speed at which people changed their clinical practice and the amount of evidence we changed it with?
Well, I think that's a complex question. I think early on, when there's no data, then you're pulling out whatever you want with the patient in front of you, trying to help them. And so I was like, let's give the guy hydroxychloroquine. I don't know if it's gonna work or not, but it's probably not going to harm them. And so people
reminds me of the vitamin C data for sepsis. It's like
all sorts of throwing all sorts of stuff at them. And I think that's fine when there's no data. You practice medicine based on your own instincts and what you think might help. And some people are more nihilist. They're like, well, we don't want to do anything. It's like, I don't think that's the case. But once you start to have data, then you change your practice. And so I think that's the key, that when there's no data, that's fine to be very experimental and you're helping one patient in front of you, which is very different from crafting a guideline or from an FDA perspective or a regulatory perspective. And so I kind of view those very differently. And so people early on that wanted to treat their patients with hydroxychloroquine, I'd say, well, participate in the clinical trial would be my first choice. If you want to do that, that's fine. But once you get data, then you need to kind of adjust. And so I think that's the key.
So what happens next in terms of Fluvoximin and eventually when did you file the approval? The approval must have been the fall of last year.
It was December. It was the emergence of Omicron. So with Fluvoximin, there were basically 3 clinical trials that I was involved with that were set up to test Fluvoximin, different doses, slightly different places. So
Stop COVID, right? Oh, no, that was a different. It was a Stop COVID.
Stop COVID. Yeah, Stop COVID 2, which kind of ran. Basically, they all kind of started in January, February of 2021, and one was in Brazil, the Together trial, and we basically via Minnesota, the COVID Out trial, we kind of ran a partner trial with the Together trial, except ours was much slower than theirs, but we did a different dose. And then the Stop COVID trial ran, it was through WashU, ran until May. And basically, they couldn't enroll any patients and kind of it was futile to continue. And so they gave up in May. And so, there was sort of a series of different clinical trials sort of ongoing to try to verify the first finding.
Right. And it showed that there was some efficacy, right?
Yeah. And so the first one that came, or I guess the second overall trial that came out was the Together trial run in multi-site sites in Brazil. And there was basically about a 25-30% reduction in hospitalizations. Depends how you define what is a hospitalization, which we can get to later. And so I think
the FDA hit you on that. I think
they did, yeah.
So we'll save that when we get to the application. Okay, so all right. So there's all this stuff you're finding, and it's promising.
Yeah, it's promising. And so life is going on, and we got monoclonal antibodies, and Delta is happening. There's a lot of Delta cases, but there's monoclonals available. vaccines available, widespread at that point. And so, the urgency, fluvoxamine is one option, but there's other options in the US as well. And so when I sort of became much more concerned was the emergence of Omicron. And so in December, mid-December of 2021, basically when Omicron emerged, we then immediately lost all of the monoclonal antibodies. And then right around the time molnupiravir had...
When you say lost all the antibodies, are you talking about the government taking over the distribution of them and then, or rescinding the EUA? I mean, can you just clarify exactly what you mean?
Yeah, so lost meaning the activity was lost. So the Regeneron monoclonal antibody, which had worked great for the last year, basically did not have any binding, didn't have any activity against Omicron. Similarly, the Eli Lilly, I forget, I can't pronounce it nowadays. I haven't pronounced it for 6 months. So the Eli Lilly,
sulfusib, no, what was it?
Bamlanivimab.
Oh yeah, BAM. Yeah,
BAM.
we just called it BAM.
BAM,
yeah.
Exactly. So the BAM one also was lost, and so the
Sofo and BAM.
yeah, exactly. Yeah. So the monoclonals were lost, and so molnupiravir data had come out, and it had gotten an EUA. And then the Paxlovid data had just come out as well, but those were not really available, even though the data had come out of like, hey, these look great, but they're not actually present. And so with the Omicron in sort of mid-December, I was like, this is a problem. Like, we really have no therapeutics. We've gone back to nothing that really works. And so there's a ton of people using Ivermectin and other things that probably, well, now we know don't really have
off-label, yeah.
Off-label use of Ivermectin, and just was, hundreds of thousands of people, per month. And so this doesn't, we need to steer people, like try to get people using something that actually has some activity.
Can I give you a, just my clinical experience? I remember right as the emergence of Omicron was happening worldwide, hadn't quite happened in the US, I felt like the rug got pulled right out under me. Yeah. I have a huge patient population that, for whatever reason, vaccine values, wasn't quick to adopt the vaccine. Many did, but I had a population that still was evaluating the data for themselves, and we relied on those antibodies, especially for the high risk. And I felt like as Omicron, and they got the numbers wrong. I don't know if you remember, they got the numbers wrong for the first 2 weeks of Omicron. They overestimated the Omicron. And I'm like, just let me give like every hospital. I remember I had a 72-year-old patient with diabetes, hypertension, coronary disease, every risk factor you can, on immunosuppressives. Husband has COVID. She has COVID. They're both going to die. And I literally called 5 heads of infectious disease at local hospital centers begging for antibodies for this couple. I mean, I have never felt so desperate in my life. I don't know if people, so that time was a troubling time for me.
It was. I mean, hospitals were full, and it was winter, and it was pretty bleak. And the case, when, the case numbers were, crazy high. And so, and it was pretty bad. And so that was the point at which like our hospitals are getting overwhelmed. And so, fluvoxamine, there's a modest benefit. It's 25, 30%, like it should be a real therapy. And that people consider, it's not your first line. If you got first line choice of other things that might be available, that's great. But like most people had nothing available to them. And so our
like
point
me.
is, and so like, are you better off with a 30% benefit or observation of 0%? And so like 30% benefit on a population scale is huge. And so it's not a perfect medicine. There's tolerability problems. And so if you can actually tolerate it, in the Together trial, it was a 2/3 reduction in ER visits and hospitalizations. And so it was even a larger benefit if you could tolerate the medicine. Not everyone can.
Yeah, coffee is a big one. That's what we learned that real quick.
Yeah, it interacts with caffeine. And so that you got to kind of give people some guidance. But, so anyway, so we thought like, hey, we should steer people in the correct direction. We'd given the data NIH guidelines, and they're like, well, we'd like to see, we'd like to, an IDSA says, well, we kind of want to see, does this really reduce deaths and ICU stays? And it's like, this is outpatient therapy. Like, you don't power a trial for mortality.
That was a time when they switched Remdesivir to outpatient with even less data, right?
Yeah.
So right around that time was
not a whole lot of data. That might have been a little bit later, but that was, Yeah, and so they jumped on the molnupiravir data, like, oh, this is great, like 30% reduction, by intention treat, and so long as you don't count the person lost follow-up, and like there's one patient different, then it all goes to heck, and you lose statistical significance, whatever. So they jumped on board, and everyone's like, oh, molnupiravir, that's great, and it's like it's the same as fluvoxamine, except fluvoxamine's $5 and that's $800, and so that's basically once molnupiravir got an EUA. We thought, the data is not as good with molnupiravir as it is for fluvoxamine, so we should apply because people need options and we need to steer people.
Yeah, but this is a big, okay, I want to like focus on this. We should apply.
Yeah.
Can you walk me through that? Because I haven't met other doctors that would do that.
Right.
Right.
So,
this is a rare thing. So, what's going on in those discussions?
Right. So, basically, for an EUA, so the FDA does have some guidance document online that says, give us, these dozen different parts of the thing. And so most people, if you're not familiar with FDA paperwork, that would be just daunting to start with. But it's just with any government organization, you just got to follow the rules. They tell you what they want, and you give them the data that they want in an organized, coherent manner. And you can do it. So you also do need a drug company to kind of you have to reference some drug company's data, and that drug company has to allow you to reference their data because it's an existing medicine. And so basically, because they collaborated on some of the trials, we basically emailed the CEO and said, hey, we need to cross-reference your ANDA letter kind of thing and get you a sign-off sort of for that. And
just so everybody knows, the generic drug company who he's probably reaching out to saying, hey, can you help us? We want to use your data as part of this application because we need a sign off from industry.
Yeah. And so because this is a $5 treatment course, generic medicine has been around forever, there's not a lot of impetus for drug companies to like go through this effort to file the paperwork because like it's a generic drug and so anyone can sell it and there's no profit margin. And so are you going to spend, hire a bunch of regulatory consultants to get this approved? And so the answer is no, because there's not a profit margin for it. And these companies, that's not their expertise. Like that's not what they do. And so basically it was a December, that was right before Christmas. It was a snowy winter holiday weekend. And like there was a ton of Omicron, so I wasn't going to go out and go socialize anywhere. And so I spent kind of 4 days, writing up this EUA application over the course of a weekend. And I sent it to some colleagues as well to proofread it and have their comments as well and get data from Ed Mills and the Together trial and some of the other trials that provided their raw data sets as well. So yeah,
why are you doing this? You're going to get no money. You're going to get nobody gave you a pat on the back. In fact, the FDA gave you the middle finger. So help me understand. You have I don't see any way David Boulware benefit.
No, there's no profit margin for me. There's no. I mean, it's my time. I'm either wasting my time or giving my time away. But the goal is to help people. And so when you think about why did you go into medicine, it's to help people. And so if I could get this EUA and get it approved such that and steer people and steer doctors the right direction towards a therapy that has a modest benefit, ultimately that's going to save lives. And so that's why I did it.
And just so anybody knows, this has never been done before. Never been done before. Okay, so you do it.
It's a little unusual, yeah.
Yeah, so you do it, and let me tell you, immediately I'm like, fuck yeah, there's somebody. Sorry, Jack, edit that one. I'm like, there's somebody out there, because I remember at that time when you submitted the EUA, I don't think most people understood. Like, it's not like a one-page, it's like hundreds of files.
150-page document and subsections and appendices. Yeah, it's a little bit.
That was like the shining light. There's Surgisphere, there's the horse paste, there's, people are grasping for straws. I have patients on one side of the political spectrum getting their fourth vaccine, lying that they didn't take it, just so they can get 2 of each one. And I have other people taking Ivermectin, off of an online consult. And I'm like, somebody, and you see the kind of the issues that are inherent at the NIH and the issues of fraud that have come up, and you're looking at the FLCC meta-analysis, and I'm thinking like, this is all BS. It's not BS data. It's just not low quality data with deserves a high degree of skepticism. And I'm like, I feel like the middle child, you know what I mean? And then come you, I'm like, holy shit, there's people out there putting in the work, you know? And I just wanted to let you know, you're my hero. No, I mean, you're laughing, but you know, you're my hero. And there's other people, Tracy, you know, there's some people who've been a shining light who we're going to interview also, I think, throughout this whole thing. People who've put in the work, no benefit to their own. So you're my hero. So what happens? You put this application together, you start working on Ivermectin trials, right? What happens?
Yeah, so Ivermectin trials are running in the background along, you know, during this time period. And so I send it to FDA and I hear crickets. Like it's like December 21st. That's right before Christmas. So okay, I give them the benefit of the doubt. They've checked out. It's a government, you know, government worker. They've gone home for the holidays. And so I literally hear nothing for about probably 2 or 3 weeks, and it's like follow-up email, like, did you receive it? Oh yeah, we're reviewing it. We'll get back to you, you know, once we reviewed it. It's like, well, you do know there's a health emergency out here with Omicron, and like hospitals are overwhelmed, and so like feel free to review it, you know, when you can get around to it.
Wait, there's a fee for the application, right?
There's not actually.
There isn't. Okay, I thought there was a fee.
For an EUA, there's not, and even if you're a nonprofit, like for a new drug application, if you're a nonprofit, there's not a fee. But
okay, got it. So no fee, but you don't have a revolving door, right? So you know your revolving door. So the industry is not going to speed things up just to work for you in 1 year. Okay, so what happens?
Yeah, so I kind of maybe about 3 weeks later, I get a follow-up email of like, well, on page 22. You know, you put that the inactive ingredients are this, but I think on this formulation it's that. And I was like, sure, okay, yes. So I submitted a correction of like it's dye number 6 instead of dye number 4 or something like that. And so the correction of the inactive ingredients. And then they asked for like a Word documents we had provided some the patient information sheet and the provider information sheet in a PDF format. They asked for the Word document, which I was like, ooh, that means they might actually want to edit it and actually like do something. Which I took as a very good sign. And so they basically had no questions whatsoever. And then another month goes by and there's some more data that comes out. And so we get the COVID out data, which provides some dose, it's a different dose than what we asked for. And so that was sort of informative. We thought it was informative to help sort of figure out that the needed dose was 100 milligrams twice a day and that the 50 milligram twice a day dose was too low and didn't have a significant benefit. And so the, you know, she provided some updates and provided a meta-analysis that was published and some other sort of stuff that was sort of coming in that once again sort of reinforced. And then they asked about 2 months in, sorry, it was about 6 weeks in, they asked for, and this was kind of when I knew we were in trouble, like, oh, this is not going well. So they asked for hospitalization only data. And so I kind of asked them, well, you know, for molnupiravir and Paxlovid, they define hospitalization as hospitalization or acute care for more than 24 hours, whether you're in a field hospital or ER being held there. And so that was kind of a definition that was used across multiple clinical trials. And they said, no, we want hospitalization only. Like, we don't care about these prolonged ER visits if you're in the ER for 24 hours or longer. Like, we want hospitalization.
What did you fear at that? What did you feel at that point? So when you got this and you knew, because you and I know what this means, right? They're playing favorites.
Correct.
Right? So how did you feel like you're out there working and there is this inherent systemic bias for whatever reason? You know, what are you thinking? I mean, you're like a science guy. You want to trust the system. You know, Surrogate happened. You know, everybody's grasping for straws. You know, Hydroxychloroquine doesn't work. You know, politicians, all of this, and then you go through this work, and they basically are like, "Screw you," you know.
Right. So they said a couple of things. Like eventually, in their sort of release letter of why they rejected it, of like, you know, progression to severe COVID, like that's not a valid endpoint. Like, so for outpatient therapy, if you start with sort of a mild cough and you like end up in the ER, you're hypoxic with low blood oxygen levels, that's kind of deterioration in my book. And preventing that, most patients and doctors would say that's a clinical benefit. If you prevent people from having to go to the emergency department with low oxygen levels and needing hospitalization and or potentially dying. And so, but they're like, well, you know, that's not really, that's not a valid endpoint. It's like, but you defined what severe COVID is. Like, this was what was used in the vaccine trials of like, preventing severe COVID. That's the whole point of vaccination. And so that was disappointing. And then
this is 3 months ago. Just so everybody knows, this is 3 months ago.
Yeah, 3 months ago. And then the context of ER visits. And so the Together trial used the term prolonged ER visits. So it wasn't like you walked in the ER and they said, "Oh, you're fine, go home." It's where then they sat on you for at least 6 hours for observation, like treating you, giving you steroids, giving you oxygen, giving you IV fluids, etc. At least 6 hours, or you're hospitalized. And so it was a composite combined endpoint,
which they do all the time in clinical trials. This is like ubiquitous to power the trials the way they need. Correct. To get a slight approval, to get a slight positive effect.
Yeah, because you want to try to do a trial with as few patients as possible. And so this was a 1,300 person trial, so it's not exactly a small trial, as opposed to like hospitalization, which might be a 2,000 person trial. And so that would take more money, more time. And this is once again a generic drug, which is based on the trials from basically donations from philanthropy. And so they had this combined endpoint of prolonged ER visits, or if they analyze the data as exactly the same as the molnupiravir and the Paxlovid trials of greater than 24 hours stay or hospitalization. Once again, statistically significant benefit, about a 25% reduction. And FDA basically ignored it and said, well, we want hospitalization only, which was really quite disappointing because it was overt hypocrisy of like, which I told them like, you are being like here, you're overtly putting this in, and this is anyway. So
hold on. I don't want to make you say something. I don't want to make you say something you don't want to say, but I want to explore this in a professional way.
Yeah.
So what's their angle? Right? They must have an angle. These guys are like regulators, and they must, what is like, if we have to suppose, is it they wanted harder endpoints for an off-label use? Like, what's the angle? If you and I were the regulators, why would we say what they're saying?
Well, it's interesting because most of most of my interactions with the FDA over the years have been really quite good. And so, like early in the pandemic, like the normal staff you work with are very professional. They're they're really good statisticians. But when you get up to like the EUA level, that's a political decision ultimately, fairly high up the food chain of FDA. And so with the hydroxychloroquine, when I was talking to the normal sort of worker bees at FDA, like, why did you give this an EUA? This is ridiculous. They're like, well, it was above my pay grade. And so I was talking like, deputy directors, and it's like, there's not that many people above you. So you're saying something. But anyway, so it does become a sort of institutional political decision.
Or when you say that, institutional culture. And what we know about the institutional culture at the FDA is very much a revolving door. Remember, we are dealing with, people who, for example, let's go to the SSRI data. It was Hughes who wrote the book, The Emperor's New Drugs, where he had to use a Freedom of Information Act to get all the negative trial data on SSRIs to do a proper meta-analysis.
Yeah.
We're dealing with the FDA that dealt with, Purdue Pharma during the opioid epidemic to give them favorable claims on their label in terms of abuse potential. So we're dealing with a government institution that has potential for systemic, like any large institution, systemic issues. Right.
There certainly can be, and I'm not an expert. I won't say I'm an expert on that. So
yeah,
I think one thing that goes against, there's several things, but one thing that goes against Fluvoximin is people don't understand the mechanism because it's not.
That's what I was going to ask you. How does it work?
Yeah, how does it work? And so this is thought to work on these sigma-1 receptors, and it basically dampens and downregulates inflammation. And so you sort of prevent the cytokine storm is the theory. But it's kind of all sort of theoretical, and you can't really measure that per se. And so that's part of the issue of like, they don't really understand it. When I went to med school, like I never heard of a sigma-1 receptor. Like, what is that? Like, it's something inside your endoplasmic reticulum, like never heard of it. Versus like an antiviral, like, kills virus, blocks virus, like virus goes down, we can measure that. And so it's much more intuitive, you know the mechanism of action. And so without a mechanism of action, I think the bar for regulatory approval is higher, which is odd because it's an existing drug. And so there's two types of EUAs. So one is for a new compound, which we've heard about, throughout the pandemic. and the other is an EUA for just the label change. And so where you can put a new
which is
label
easier,
on,
which is, should be easier. Yeah.
You've got all the existing safety data. And so, you know, you got decades of safety data with this medicine. And so you would think, oh, a label change, the barrier for needing data would be less, because normally you need basically 2 positive studies to get a label change. And so we had 2 positive studies for a label change.
You had 3, right?
I would say, well, Together trial and the initial WashU trial.
Right. Got it.
And so those, and so we had like, hey, this is the normal regulatory, thing for just a label change in general. So you think this is a lower barrier because it's an existing medicine. But they, obviously they disagreed.
So help me understand, we've seen Paxlovid approved with kind of surrogate endpoints or maybe flimsier endpoints. We saw other treatments approved. And, since then we've seen kind of the vaccine discussed with even like more of the surrogate endpoints than that. Right now we're just showing that does it make antibodies without necessarily knowing what it does in the end. So do you think that it's do you think it's just the fact that it's because in the case of, pediatric vaccination, they accepted a surrogate endpoint of antibody production, and there was absolutely 0 clinical endpoints, if I'm not mistaken.
Yeah, there was some, but it was a little weak, and depends how you analyze it. But that I think is actually fairly standard, where you've gotten the proof of concept in the adult population, and so and the adolescent population. So then it's just really about, do you make the same antibody levels? And so that's been done with other vaccines in the past. So that is kind of a standard practice, because let's say you have a new vaccine for XYZ disease. It's really, you don't need to necessarily repeat every single phase 3 trial in every population. It's more about safety. And then if it's the antibody that protects you, so that's a little bit more standard. The Paxlovid, so just for clarification, the Paxlovid has a great benefit in the unvaccinated or immunocompromised population, but in the not that population, then the data is quite murky of sort of minimal benefit.
Yeah, we've used Paxlovid since approval on several occasions.
The molnupiravir data is a little bit more shaky and probably is going to be very minimal. That data is really quite shaky.
So these are all EUAs. These are all emergency use approval.
Correct.
So now you get this feedback from them. When did you get the rejection letter?
It was fairly recently. So this is it's mid-June when we're speaking. So that was it mid-May, I guess, about a month ago or so.
Right.
And they emailed me. It was a very polite email saying, there's a lot of interest in this medicine, and we thought we should release why we declined your EUA.
And that's also that they didn't have to do that.
They didn't have to do that.
So that's like an olive branch, at least. Like, hey, look, because they can just say, hey, we rejected, and
correct.
That's it.
Yeah, they're not obligated to provide any feedback. And so they did a very comprehensive review. It's 20 pages or so. And they kind of went through the pros and cons and what the data was, and it was clear that they were kind of, they weren't using the same definitions that were used for other drugs, for the Paxlovid and molnupiravir. So I read it and I emailed them back. It's like, are you sure you want to release this? Because this makes FDA look really bad. Like you're overtly, like, people always complain about how there's the double standard for big pharma versus little pharma.
Yeah, I'm just doing it right now. The systemic
bias.
You're
putting it in writing. Like, are you sure you want to do this? Because I generally, I don't want to, I give, try to help people out. And so, like, are you sure you want to do this? And so they're like, oh yeah, we're positive. That's it's like, okay. So I said, do you want any feedback on this critique? Because your critique is not totally logical, and there's some logic holes in it. And they said, no, we don't accept feedback. It's like, well, you're gonna get feedback regardless, but it's gonna be in the public domain.
Got it. So you get that back, and that must be a huge blow. I mean, when I saw that, it wasn't unexpected.
No. By that point, it was like, it's been months. And Omicron has sort of crested and come down, and so it was less.
What about oversight? Like, how would 3 docs manage oversight moving forward? They probably wanted some sort of oversight, which you couldn't provide, I'm guessing, right? Like, as in post-marketing oversight, right?
Possibly, yeah. But the pharmaceutical companies, we had that. We had a plan for that.
Okay.
Maybe it wasn't a very good plan, but they didn't question that. That wasn't one of the issues. But
Got it. Okay, so this got to be discouraging.
A little bit, but
You
I guess I'm an
expected.
optimistic person. If you get discouraged, you don't, nothing ever changes.
Got it.
So I think,
and then you get two interesting trials. So now so during that time, everybody's grasping to ivermectin, and we're seeing trial after trial come out negative of ivermectin. I remember the ITEC trial was the first one that really said, like, this is it. Like, I was already cautious on the meta-analysis from the, FLCCC and not to say that, I didn't read and I like these people's voices, the contrarian thinking, the vocal thinking, willing to look at observational data. But, ITEC came out, which was a pretty well done trial in Malaysia. And then, I think it was your trial that came out. That was the, it was online. Yeah, the Together trial was online, right around the same time.
About the same time, yeah.
Yeah, and I know it just recently got approved. Recently got published, maybe a month ago or so. But now we had two fairly well done studies. Although some people argue that the dose wasn't high enough for ivermectin in obese people and this and that. And then there was another trial, which that was there was one more trial. Am I blanking or did I get it right?
Active 6.
Yeah, Active 6, right? Which you're also part of.
Also part of that, yeah.
So help me understand ivermectin, because that has mechanistic data.
It has mechanistic data, and so the first paper came out in like April of 2020, and so I knew ivermectin. I prescribed it a lot as an antiparasitic drug for strongyloides and other stuff. I've written about it a lot for that. And the first thing I did was figure out they had some level in a petri dish where it inhibits the virus. And so the first thing I did was figure out, like, what's that dose, and can you actually achieve that in humans? So you kind of do the calculation. They present it in millimoles versus, which is not what you measure the drug in, and so you got to do some mathematical conversion. But anyway, and you figured out, like, this is 50 times higher than the normal drug dose you're ever going to achieve in humans, and so this is way too high. And this happens all the time, where in a petri dish you can look, oh, look, XYZ drug works against whatever. But it's like, can you actually get a drug concentration that high to actually achieve it? And so I looked at it in April of 2020 and said, oh, this is interesting. Did the math. It took about 15 minutes. It's like, there's no way this is going to work. It's like 50 to 100 times too high a concentration needed. And so I kind of just put it away and said, well, this, whatever, doesn't work. But there was a lot of it.
Not that it doesn't work. It's not clinically feasible.
It just doesn't. It doesn't theoretically make sense to pursue this as an option.
Yeah.
But there's clearly a lot of promotion of it and a lot of people selling it, and there's only like two U.S. approved manufacturers, and so therefore the price is kind of high, and so you can make money off the drug, and there's a limited supply, and so and so there's a ton of use. And so this is why one of the things, like, hey, we should actually study this because it's being used, it's being promoted, but the data is all over the place. There's some clearly fraudulent data. There's some stuff that, well, maybe this does have a benefit. And so I was relatively open-minded. Certainly in people in low and middle income countries, in strongyloides endemic regions, there's basically a parasite called Strongyloides stercoralis. And so if you give someone steroids, like we do for COVID, for severe COVID, the worms kind of go nuts and they crawl throughout the body, termed hyperinfection, and it's kind of a terrible way to die, I guess. But it's like sort of, Star Trek II Wrath of Khan kind of thing.
So you're trying to come up with why it worked in the third world.
Why it worked. So there's a theoretical reason why it could very well work in low and middle income countries in hospitalized patients, but that doesn't mean it necessarily is going to work in the U.S. And in the outpatient setting when you're not giving people steroids, and so it's worth a clinical trial. And so at the time when like Active 6 was starting the planning, which was like February of 2021, we talked to a lot of different people about the dose and sort of even the FLCC people, other sort of serious doctors and around the world kind of thinking about dosing and what the other prior phase 1 and phase 2 studies have been done in malaria and other diseases, looking sort of at higher doses. And so, that was sort of the dose for Active 6, which was very similar to Together and similar to the COVID Out, was all sort of around this 400 micrograms per kilo dosing.
Yeah. So, and basically all three trials were negative, right? Were basically negative. Well, I shouldn't say negative. No, what I saw was possibly some reduction of duration of symptoms.
Yeah, and so I think
how much else?
Yeah, are we now debating, is there a zero effect or a minimally beneficial effect? As far as, in active 6 with ER visits and hospitalizations, there was not a reduction. There was a half a day, about actually 11 hours and 46-minute reduction in average duration of symptoms.
Did you say 11 minutes?
No, 11 hours and 46
11
minutes.
hours. Yeah, okay, got it.
So if you say, well, that 11 hours, what could you do in that? I don't know. If you're sick for, 10 and a half days versus 11 days, or I guess 11 days versus 11 and a half days, is that a big difference?
And I'm sure subpopulations didn't reveal much, right?
Yeah, and so there's some, you can dive down into the subgroup analysis. There is one sort of, small subgroup analysis, but it's an interesting one of people with severe symptoms at baseline. They seem to have faster recovery. So maybe it's 16 hours or it's 18 hours faster, but it's statistically different. Is that a real effect?
But basically enough for you to say this isn't worth pursuing further.
Yeah, this is not a real sort of viable therapy. And so if you put that in combination with the Together trial and the ITEC trial, and there's an Iranian study that just came out yesterday. There's also the COVID out trial, which has not been released as yet. There's a bunch of large double-blind randomized placebo-controlled trials that say at this dose and this duration, there's not really an effect that's clinically meaningful. People can disagree, that's fine, and say, well, half a day duration, that's really important. That's if you think
it's not really important for me personally. I think that the appeal to ivermectin was people saw fraud and problems in the regulatory environment that we just talked about, and they turned to something that wasn't industry guided,
and you could buy it over the counter at your local, local Tractor Supply store. You didn't need a doctor's prescription, or you could go online and people would sell it to you. So you can kind of get, avoid the
losing your job because you're not vaccinated. Avoid that, right?
Yeah. So, and I think some of the stuff that FDA did was not very helpful. Of vilifying people and like trying to make fun of them with horse jokes is not helpful. I think that most people would agree with that. Maybe not everybody. Some people like to make fun of people. That's fine. You can have jokes. But like, if you're trying to do a public health thing, like making fun of your target audience where you're trying to engage them, is not an effective sort of way to public health to do a public health message. And so I think that was kind of turning this into a joke, or once again making this political. Was not very helpful.
Yeah. So okay. So now here we are, right? You've worked. We went through the iterations of hydroxychloroquine. We found benefit to steroids, and they're widely adopted now, and certainly recommended in most guidelines in severe disease. You tried to get Fluvox approved. They basically shot you down. Instead, we're using, I don't know how much it's been in clinical use, but I know it's approved at least for high-risk outpatient use. You can inject Remdesivir, I believe. Am I wrong on that?
Yeah, that's true. You can get 3 sequential days of IV Remdesivir.
Right.
Our institution looked at this and said, there's no, just from a nursing staffing perspective, labor, it's just too labor intensive. And so we couldn't do it. And so it was something that one-time monoclonal infusion, like that's easy, or a subcutaneous injection, that's easy to do. But if you're doing 3 days of IV therapy, that's just really hard to do. It's very labor intensive.
So here we are at the end of all this, and, now we have Paxlovid, now we have all this, but has your opinion of our field changed over the last, it's been a year, and I want to kind of wrap this up because I want to be mindful of your time. I'm sure you have things to do, but has your opinion of our, the regulatory environment, our central planners, seeing the kind of the surrogate endpoints accepted here and there, and then seeing the other side, people desperate for, alternative treatments and being maybe excessively hopeful. Has it changed your view of our field? Because I don't know, am I the only one who feels, very few people have given me a feeling of shining hope,
Yeah, I think I'm certainly a little disappointed. I think particularly some of the regulatory stuff, obviously, and some of the guideline panels, because it seems like the objective is a drug has to be perfect. And so Paxlovid for immunocompromised or high risk people, great, huge benefit for reducing hospitalizations, no benefit for actual symptom reduction that we're aware of, or symptom duration. But like, hey, that's great. But like, doctors need options and patients want options, because in our health system, about 15% of people have some contraindication to Paxlovid. And so you need a plan B, you need a plan C. And so instead of just having like, it's recommended or it's not recommended, it really should be a tiered structure of like, well, here's this and here's this and here are your options, and have more of a tiered structure based on availability and patient preference. Because some people don't want Paxlovid, it's a new drug, or molnupiravir, it might be, kind of mutagenic, it might change your, your genes. Like, oh, that's not so great. Or you're a pregnant woman, like, you're not going to do that medicine. And so having options, I think, is something that that hasn't been articulated as well. Of like, well, we want more data. We want more data. We want gold-plated, gold standard data. But in a pandemic with emerging data, sometimes you got to make choices, and you.
Well, they want gold level data from you, not from industry.
Industry is like, well, one study, barely statistically significant, it's okay. And I think the thing is, people don't want to be wrong. They don't want to endorse a medicine and say, yep, that works. We're going to put it in our guidelines, and then have to retract it. They'd much rather sort of say, well, we need more data. We need more data. We need more data, which is fair. But in that case, you have to look at what the consequences are. And so if people are using unproven therapies that don't work, then that's not helpful in the big picture. And so I think that was a mistake that was made that really has not been super well rectified. WHO is doing the same thing of like, well, we want Paxlovid for everywhere in the world. That's great, but that's not going to happen because there's a very limited supply. And so, like, WHO doesn't endorse Fluvoximin. Well, we want some more studies. We want more data. So most people in low and middle income countries have no access to any treatment, where this is a low cost, inexpensive treatment. It could be used. But like, WHO is like, we want Paxlovid for everybody. Like so people are in this weird, fantasy land sometimes.
I want to part with these last words, because I think this is your thoughts on this last thing. So here we have so many people don't follow the AdComs. I've watched every AdCom, looked at all the documents. I mean, I don't know any other. I don't think I would have done this had I not been exposed to it prior to med school. But so, an FDA deputy, resigned, right? Somebody who's part of the advisory committees. The advisory committees were at some point disbanded, recently brought back. And very recently, they unanimously approved vaccination for 6 months to 5 years under EUA. What do you think that says about the pressures on our central planners, right? That they kind of halted the way that they typically do things. People have resigned. What do you think it says about, you're an MPH, the difficulty of public health. You know how hard it is to be right in their position. It's almost impossible.
Yeah, I think it's impossible, and people don't want to be wrong. No one wants to be wrong. But clearly, I mean, when you make decisions based on the data you have, but you can always revisit those decisions. And I think that's where, sometimes people are gun shy, where they can. I think you can say they swing either both ways, where they're either too restrictive and they want more data, or they're too lenient and they're approving things that we really want more safety data on or more efficacy data on. You take the Alzheimer's, drug, for instance, of, where like, is there any data this works at all?
So he's talking about the Biogen drug that was approved.
Yeah.
I don't know, with very flimsy data.
Yeah, that's not my field, but kind of just the general gestalt is like, it's unclear if it works at all. And so that's one extreme where there's very little, in others where they just want too much data. And so there's always intrinsic balance of safety versus efficacy and how much data you need for each one of those. And it depends on the population. And so in kids, kids, we'd like a lot of safety data. And so small trials of 5,000 kids certainly is not going to identify rare side effects that occur 1 in 10,000 or 1 in 30,000 people. But it's a starting point. And so I think that's, if there's, phase 4, basically post-marketing surveillance, it's actually robust and actually good, then you'll identify, follow-up safety issues, and eventually that will be corrected. And so I think like the J&J vaccine is one case of that, where there was concern about thromboembolic events, and so that happened. That was kind of discovered relatively early, maybe not early enough, and basically no one sort of uses that vaccine anymore.
So you're talking about the 1 in 50 to 100,000 people that develop this clotting bleeding issue.
Yeah.
That what strikes me is that Europe, kind of shifted 6 months prior to the United States, again,
Correct.
favoring industry. And then the other thing that I noticed was a lot of Europe has already shifted away. There's a clear signal in our young men, particularly age 5 to 15, the myocarditis. Maybe as low as
teenagers, yeah.
Yeah, 1 in 5,000, maybe as high as as high as 1 in 5,000, maybe as low as 1 in 50,000. Depends on who you ask and what endpoints you look at. But Europe's already moved away from the Moderna vaccine, which appears to be minimally worse. And so I wonder, is our regulatory environment a little too favoring and cushy with industry? And why is it that Europe is quicker to act? And, that's just, I'm not going to answer that question. I'm not going to even let you answer that question. I'm going to say, thank you so much. This means a lot to me personally. I consider you a personal hero of mine. And many people may not understand why, but it's because you put in the work, right? You put in the work. And whereas a lot of people have just clung to their kind of idols and politicized it. Somebody, who's, nobody has done this. No, and I don't think people understand it. It's a small little thing that I'm getting hung up on, but to me, it says, man, this guy is somebody to watch for. So I want to thank you for what you've done, the research you've done, and thanks for helping empower docs like me to practice clinical medicine. And thanks for coming today.
No, it's a pleasure to be on today on your podcast, and I think there's one more really important clinical trial that's going to come out, and that's the COVID out trial, which I'm very excited about. I'm sworn to secrecy on what the results are, but hopefully it will be out very soon. And I think it'll be very interesting, and I'm very curious of what people's reactions will be to that trial.
What was the intervention? I'm just curious.
So there were 3 interventions, so it makes it so then you don't know which one it was. So there's Ivermectin, there's low-dose Fluvoximin, and there's metformin. And so and there's a factorial design trial, and so it's a little complicated.
I'm gonna guess metformin beat both of them, and Fluvoximin came in second, and Ivermectin came third. That's my guess.
Okay. We'll see how good your guesses are. So
yeah, and when does it?
Unclear. So it's in the works, and it's with the journal and unclear timeline for being released to the public. But hopefully in the next month or so. I would hope so.
Awesome. Can't wait to see. I cannot wait. Thank you so much.
Great. All right. Take care. Thank you for the invite.